I am a faculty investigator at the HudsonAlpha Institute for Biotechnology, located in Huntsville, Alabama. We're a nonprofit genetics and genomics research center, and we're all dedicated to the use of genetic and genomic technologies to improve human health and welfare. And today I'm excited to talk to you about some of the work we've been doing recently focused on long-read genome sequencing for the diagnosis of neurodevelopmental disorders. So just to set the stage in terms of the kinds of phenotypes we're talking about here, neurodevelopmental disorders are kind of an umbrella term that captures, it's approximately 1 to 3% of children that are born with symptoms including things like intellectual disabilities, developmental delays, dysmorphic features, autism, seizures, and other kinds of problems related to growth and development that can lead to a whole range of different cognitive and physical challenges. And the vast majority of these, or at least a very large fraction of these, have genetic causes. In fact, tend to be rare genetic causes. And because they're rare genetic causes, in order to find a correct diagnosis, you have to find those rare genetic causes. And so and an accurate diagnosis is in turn really essential to give families the best possible counseling about what caused the problem, what to expect in the future, of course, to treat the affected children in the most effective way in terms of whether that's pharmaceuticals or educational or counseling or social connection to other families, that sort of thing. So, and of course, in the long haul, we really need to understand root genetic causes in terms of developing the most effective treatments, doing research on effective treatments in the future. So that's really the focus here today, is on the discovery of these rare genetic factors that can underlie neurodevelopmental conditions in kids. So we've been doing this for quite a long time at this point, so HudsonAlpha in general. And so we've partnered with clinics and hospitals throughout, especially Alabama, but also really across the Southeast, to identify children that had an unexplained neurodevelopmental disorder. So our primary partners are, our first partner, in fact, was Martina Bebin, a really wonderful UAB-affiliated neurologist that performs most of her work in Huntsville. That was part of a project called, we were part of an NHGRI program called CSER. We work with Alabama state agency that helps a family of children with special needs called CRS, and they have clinics all across Alabama that we can recruit through. We currently have a grant as part of a CSER renewal called SouthSeq, where we're sequencing babies that are in a neonatal intensive care unit at 5 different hospitals across the South that are indicated here. And essentially, we're looking for babies that have signs of a rare genetic condition. That's the explanation for why they're in the hospital. We've worked with something called the Alabama Genomic Health Initiative, or AGHI. This is a state-funded collaboration between UAB and HudsonAlpha that has a variety of genomic medicine goals, including rare disease diagnosis. And our sort of standard pipeline or process, historically at least, has been Illumina genome sequencing. So this is a standard generate short reads to about a 30x mean coverage via Illumina, and then perform variant interpretation and assessment. We started before the ACMG, the American College of Medical Genetics standards, were developed, but we now adopt those ACMG standards for weighting evidence and assigning terms in terms of what the likelihood of a variant being causally connected to a phenotype that we're interested in. So to summarize, a whole bunch of work over these past close to 10 years we've been doing this. So we've sequenced close to 1,400 probands. It's probably a little bit over that today. We often do trio sequencing, so that's why it's a little over 2,800 total individuals, because that includes, in many cases, we sequence parents. And you can see a breakdown here, this pie chart that shows the breakdown of pathogenic, likely pathogenic, VUS, and then no returnables. So there are about 370 probands, so a little over a quarter have a path or likely path, and we sort of tend to lump those together because those are often the kinds of the variants that have the kind of confidence attached to them that leads to allowing a clinician to make an actual diagnosis. So we have a number of genes that we see over and over again, things like Dravet syndrome and Rett syndrome, which are among the more common rare diseases. It's a little bit of a weird way to say that, but among the more probably over 200 distinct genes. So these are individually quite rare conditions, even though they add up to a collectively quite common set of conditions. So you can see that the breakdown here, most of these, a slight majority are de novo, but of course we also see recessive and dominant and occasional X-linked variants. We also find lots of VUSs, so over 200 up to this point. And these are mostly de novo variants in conserved genes that we just don't understand. And I'm not going to talk about that today, but that's a major focus of what we do, is try to understand which of those VUSs are likely to turn into, because we have seen many examples, but of course you have to do the work to prove that any individual VUS is actually pathogenic. But today what I really want to focus on is the fact that we fail, frankly, in most of the children that we sequence. We just don't find anything. And that's really the focus here today, is on understanding how we can do a better job at diagnosing children in this currently undiagnosed bucket. And so the big picture, and this fits into a broader set of research from lots of groups, is that there are many patients that have severe early onset symptoms that really suggest a large effect genetic variant based upon just the nature of their presentation. There is also, of course, a large effort to look at Mendelian disease more broadly. So this isn't just neurodevelopmental disorders, just lots of conditions that are either suspected to be Mendelian or in fact known to be Mendelian via linkage or other kinds of family assessments. And even in those families, you often fail to find a causal variant. And so the bottom line is that we believe there are lots of highly penetrant variants out there that we're just not seeing. And as a result of that, if we can do a better job at finding variants and finding those causal variants that are underlying these Mendelian disorders, that this could unlock a whole bunch of both research discoveries that are important and also obviously clinically relevant, translationally important findings. So today I want to talk about our efforts to do this with long-read genome sequencing. So this project I'll talk about is the collaboration with Jane Grimwood and Jeremy Schmutz, who are colleagues, faculty members here at Hudson-Alpha. And for a long time, they have been they currently are and have been for a long time, directors of what's called the Genome Sequencing Center at Hudson-Alpha. They're primarily funded by the Department of Energy, but other sources as well, and they have done loads of work on de novo assemblies of plant genomes. So they often joke that human genomes are actually quite simple compared to a lot of the plants that they've looked at. And so basically their focus has been on building these reference maps for these complicated large genomes. And as part of that, they have lots of expertise in infrastructure for sequencing and assembly, obviously, but that includes being beta testers for a platform from Pacific Biosciences called Circular Consensus Sequencing, or HiFi. So they had this installed about 2 years ago. And just to give you a sense for how HiFi reads work, you start with large molecules. Obviously, we're trying to get long reads, so you try to get molecules that are 10, 20, 30, 50 kilobases long. This process includes ligating these adapters that end up circularizing those molecules. And that's really the key to making this work, is that circle structure, so that when the molecules get sequenced, the polymerase can actually loop over and over again over the same fragment. And what this does is it overcomes the traditional downside to long reads, which was low accuracy at the base pair level. So you would often get something like 15, 20, 25% error rates at any given base call. But when you sequence the same base over and over again, those error rates smooth out. And the result is that you get reads that are both long. So you can get reads that are 10, 20, 30, 50 kilobases long, but they're accurate at the base pair level. And in fact, you can see reads that are essentially at Illumina base pair accuracy, so greater than 99% base pair accuracies as a result of this redundant sequencing approach. And I say this figure is from a paper published by PacBio on the technology. And so we decided to work with Jane and Jeremy, and we worked with them to generate CCS genome data on 6 children and their parents that we had previously worked with and yet still had an unexplained neurodevelopmental disorder. You can see here just a breakdown, a very high-level overview of the children that we looked at here. These symptoms that you're looking at here are fairly typical: seizures, dysmorphia, intellectual disabilities, like I mentioned before. You can see all of these children had, and I'm just giving you a small snippet here of the previous testing that had been done. So all had microarrays. We had done either genomes or exomes on all of these children. And this is not to describe all the different other kinds of chemical and any kinds of testing that had been done. And what we went ahead and generated was we aimed for about 30x coverage on the proband, and then we generated lower for the sake of efficiency. We generated lower but still fairly decent coverage, so something like 15 to 20x on average for the parents, with the idea being that all the variants that we call in the child, that we could use the lower or medium coverage data from the parents to determine whether those alleles have been inherited or not. So to give a quick sort of process overview, we took a number, and this really was a long time to develop sort of pipelines and procedures that worked. We performed a set of analyses where we took the reads and just aligned them to HD38, so treated them much the same way that you would treat Illumina reads, and then performed variant calling via deep variant here. We tried a number of different algorithms. We performed structural variant calling using, again, a variety of approaches, but primarily we focused on this PBMM alignment and PBSV caller, but I'll talk about other things as well. Ultimately, we called about 7 million sort of standard variants, so SNVs and indels, 7 million per trio, so reasonably close to expectation. And really, a really exciting result here is that there was only about 89 high-quality de novo SNVs and indels per proband. And just to put that in perspective, the biological expectation is something on the order of something like 70. A given child is on average has about 70 de novo SNVs or indels, or somewhere in that vicinity. So that's reasonably close to what the expectation is. And unfortunately, what you often see in standard approaches is several fold higher than that. You often see multiple hundreds of de novo calls for various technical reasons when you look at short read data. So this was an encouraging sign that we were getting pretty high quality de novo calls, which are obviously very important to neurodevelopmental conditions. We also generated de novo assemblies, and this is something that is really uniquely powered by having long read data that you just can't do very well with short reads. We used an approach called Hi-Fi ASM, and what's nifty about Hi-Fi ASM is that this is done in a trio-aware fashion. So you can take the information from each proband and use the parental genotype to actually build and phase contigs so that you can separate out the maternal chromosomes from the paternal chromosomes and assemble them separately in each proband. And just to give a rough overview of what that looks like, it's really remarkable how good the assemblies can be. So this is from the Hi-Fi ASM assemblies, where we're looking at within each of the 6 probands here, articulated on the x-axis. And what we're able to do is assign, as I mentioned, maternal and paternal chromosomes, and it's essentially treat their assemblies separately. And you can see we're getting N50s here. So these are, the length of contigs in which 50% of all the bases are assembled into, over 12 megabases for all cases. Again, both maternally and paternally inherited chromosomes. And in some cases, we're getting N50s in the 35 plus megabase range. So this is effectively chromosome arm scale pieces that we're getting just from the CCS and Hi-Fi ASM approach. So really exciting data at that high scale contiguity level. But as I mentioned, our focus is really on finding rare variants driving Mendelian disease. So I want to walk through a couple of stories that we found within these families that we looked at. The first is we found this interesting set of reads, and I'll talk about in a second, near a gene called CDKL5. So you can see on the bottom, this is a map of an annotation of CDKL5. This is exon 3 we're looking at in particular. So you're looking at something like 500 or so bases here. And what this graph is showing is from an IGV, so this is a standard viewer to look at alignment of reads to the reference, where we have reads from mom on the bottom and dad in the middle, and we're not showing all of their reads in this case, we're just truncating that. But we're showing you mostly reads from the proband in this case. And in an IGV image, the gray areas are stretches of individual reads that match to the reference, and the multicolored areas, so the red and green and blue, are bases in those reads that don't match to the reference. And so you can see here, in and around this exon 3 of CDKL5, we have mom and dad look normal, right? So we have, I'm not showing you all of the reads, but they all look like that gray stretch solid across the entire region here. But you can see in the proband, while we do have reads that appear to be normal across the entire region, you also have these two sets of reads that have these breakpoints in them where they transition from matching the reference to having a whole bunch of mismatches. And so they have stretches that are indicative of something different happening in the proband. And you can see we've marked with the black triangles there the breakpoints inside these reads of where this transition occurs from normal read to mismatched non-aligning read. And of course, so this suggests that there's something going on here, but it doesn't really tell you what the nature of the variant is. But this is one of the power of looking at an assembly. So here I'm showing you one way to visualize how assemblies compare to each other, which is a dot plot. And basically what this is showing here is comparisons of the assembled config from the proband against the assembled config from the reference, from HD38. And in regions where they're the same, you can see that the main diagonal here are these stretches where the reference config and the proband config match up identically. And see, for the most part, we're looking here at a chunk of chromosome X. For the most part, the child and HD38 are the same. However, if you zoom in here and look, you can see that there's a gap in the alignment here. So this is a chunk of sequence that appears to be present in the proband's config that doesn't match up to the reference assembly. Although if you can see just to the right side of that, there's a little circle, so at least a chunk of that appears to match to a downstream segment of the reference, which I'll show you in a second. So what this is telling us here is that within CDKL5, within this stretch of sequence, we have a proband insertion that appears to bear at least partial similarity to a downstream stretch of HD38. And so when you go through the work to resolve what this sequence is, it turns out to be an L1 mediated insertion that is somewhat complicated. So L1, to start with, is a class of mobile element that is still active in human genomes and can still give rise to new mutations in human beings. So this is mediated by an L1 jumping event. So essentially an L1 a mobile element insertion, it makes a copy of itself and reinserts. But as part of what this L1 did when it jumped is it dragged, so you can see that in the blue, this is a 7 kilobase insertion, almost 7 kilobases. Part of it is matching an L1 perfectly, so this is the part that tells you it's mediated by a brand new L1 hop. But what it also did is it brought a segment from the downstream gene called PPF1, an intronic segment from that downstream gene, and combined that 2.5 kb of downstream segmental duplication combined with the L1 lead to the 7 kilobase insertion. But one important feature of L1 events is that they leave what's called a target site duplication. So where they insert, they actually make a second copy of a short stretch of sequence. And in this case, what I'm showing you at the top here, the target site duplication is just over 100 nucleotides long, but it includes the entirety of CDKL5 exon 3, which is 35 base pairs long. And when you look inside the proband's RNA, so it's a very faint band, but you can see it. So here we're looking at an RNA gel here of proband compared to mom and dad, and in that little white asterisk is a band that's shifted up. So that when you cut that out in sequence, that is in fact RNAs that include 2 copies of CDKL5. So that's CDKL5 mRNAs that include an extra copy of exon 3. And because it's 35 nucleotides long, it leads to a frame shift, and this essentially converts it to an expected loss of function variant, and loss of function alleles in this gene are well established as being associated with this developmental and epileptic encephalopathy, which matches up very well with the proband's phenotype, and so we've considered this likely path. So effectively, this is a diagnostic variant for this family. So for the next proband I want to talk about is a little more complicated, but the same basic picture here. We're going to be looking at config comparison. So we're looking at assemblies of, in this case, chromosome 7, or at least what we're calling derived chromosome 7 for the proband, and a derived chromosome 9. And you can see here that we're looking at the chromosome 7 and chromosome 9 configs in the child and comparing that to 7 and 9 in HD38 along the top here, the x-axis. And what you can see is that there's stretches of the child's chromosome 7 that match to the reference chromosome 7, but there's also this interleave segments where you have 7 aligning to 9, and in the bottom here you also have child's chromosome 9 aligning to both HD38 chromosome 9, but also HD38 chromosome 7. And so there's also this little spot here in red which will show us. So basically what we're looking at here is it looks like evidence for translocations of 7 and 9 into one another, with potentially, you can see this little bit where the diagonal is actually flipped on its axis, so it looks upside down or inverted. And we'll look at the in this case, again, we've phased all these so we know that in this case, the maternally inherited chromosomes appear to have the normal structure. The paternally inherited chromosomes are the ones that have this unusual translocation. So you can see we have this derived chromosome 7, which has a chunk of about 3.5 megabase of chromosome 9 inserted. And you see the reciprocal event here where we have a derived 9 that has a large chunk of chromosome 7 inserted. And this little red piece has turned out to be a 240 kilobase inversion, where at one end of this translocation you have this inversion. And I only point that out because it's obviously part of the structure and understanding what's happening here, but it also really threw us for a loop to try and figure out. This was a really challenging region to ultimately assemble, but it does illustrate the power of where you can eventually get to with these kind of data. What was also interesting about this proband is there was a second series of rearrangements, in this case on chromosome 6. So again, I'm showing you derived chromosome 6 in the proband here against reference. In this case, it's all internal to chromosome 6. But one thing you'll notice is that we have this very large inversion here where a large segment of chromosome 6 is actually inverted relative to reference. But you also have this stretch highlighted in this red circle here where you have an extensive amount of rearrangement. And this is what we believe is a result of chromothripsis, where a small region, well, small, it's about something like 10 megabases, essentially shatters and reassembles, but it reassembles in not necessarily the right order. So to spare a lot of the mechanics of how we had to drill through that, and Susan Hyde, who did a lot of this, did an amazing amount of work to reassemble that. So what the derived chromosome 6 here looks like is we have this large inversion that encompasses most of chromosome 6. And then at one end here, we have this 10 megabase segment where you have, so here we're showing you what the reference chromosome looks like, so A through H. And you can see we have this significantly shuffled set of, these are just different segments that range from a few hundred kilobases to a megabase in size, where you have a completely different order. And in some cases, those individual pieces are inverted relative to one another. So really a sort of radically altered structure within this locus. So to summarize here, we've generated long-read genome sequence and assemblies from 6 trios with an unexplained neurodevelopmental disorder. The assemblies were really remarkable considering how it's essentially one sequencing run and generation of data. So this is nearly HD38 levels of contiguity. Obviously HD38 has a lot, it's higher quality, but we're approaching that level size rate of contiguity. And we can get those phased into maternally and paternally inherited segments. And it's really remarkable to think about doing this with essentially a single sequencing kind of experiment. I didn't have time to talk about it today, but we assessed variant detection across a whole range of variant types, including SVs, which I showed you some examples, but lots of mobile element insertions, low complexity repeats. We found an extensive amount, in fact, of even protein altering variants, so SNVs and indels, in regions that have low mappability of the reference. So these are areas where short reads just can't align accurately, but long reads can get through it just fine. And you find variants that, in fact, could be potentially impactful. So essentially, you're getting a much larger or greater fraction of the reference genome is accessible via these long reads than what you see from short reads. And really, for me, the exciting part of this is that, in fact, 2 of 6 probands that we looked at had variants that we had completely missed by previous testing, including short read genomes, but that are, in fact, likely pathogenic and that lead directly to a clinically relevant diagnosis. So obviously, the numbers are too small to say, we're not saying that the ultimate success rate is going to be 1/3, but it certainly is consistent with this being a pretty substantial yield from long read genomes to previously unexplained cases. And of course, there's no evidence that long reads, there's basically nothing that long reads won't see that short reads will. So you get the entire spectrum of normally detected SNVs and indels, but you also get this additional fraction of variation that is inaccessible. So we're really excited about this, and I'm sure other groups are as well, that as costs decline, we've already seen over the past couple of years that costs and efficiencies of this process have really improved dramatically, and we certainly would expect that to happen over the next couple of years as well. That this should be, this will become a very powerful tool both for clinical diagnosis and for rare disease genetic research discovery. And with that, I would like to thank, in particular, all the families that have participated in research with us and shared their samples and information and worked with us. And I'd like to point out, I've already talked about Jane and Jeremy and Martina and the other collaborators that we have the pleasure to work with. Susan Hyatt and James Lawler really were leaders of doing this, and I'm up here talking, but they put in a huge amount of work to figure out all these complicated structures, and so it's a testament to their effort and work in that regard. And with that, I'll say thank you and close.